Good morning, students. So, in this class we are going to discuss bioremediation of pollutants. So, the process of removal of environmental pollutants by microorganisms. So, the removal of environmental pollutants by microorganisms is called as bioremediation. So, bioremediation can be carried out in two ways. One is in situ bioremediation, and ex situ bioremediation. So, there are two categories of bioremediations: in situ bioremediation and ex situ bioremediation. So, in situ means within the place, So, where the pollutants are present. So, removal of pollutants where it is generated, that is within the place, that is called as in situ bioremediation. So, it is of two categories: intrinsic in situ bioremediation, intrinsic in situ bioremediation. So, use of capabilities, innate capabilities of microorganisms in order to degrade the pollutants, that is called as intrinsic in situ bioremediation. So, what are the intrinsic capabilities that are having within the microorganisms will be used to remove the pollutants, that is called as intrinsic in situ bioremediation. And the next one is engineered in situ bioremediation. So, it is a just modification of physico-chemical properties such as pH, nutrient availability, pH, nutrient availability, oxygen supply. So, these are the modifications that are giving to the microorganisms. So, we are modifying the physico-chemical properties of microorganisms so that it can enhance the capability of degradation of pollutants, that is called as engineered in situ bioremediation. Next, coming to the ex situ bioremediation. So, it is taking up pollutants from one place to taking up pollutants from one place to other selected area to in order to degrade the pollutants, that is called as ex situ bioremediation. So, these are the two categories of bioremediation. So, the removal of environmental pollutants by microorganisms, by the use of microorganisms, that is called as bioremediation. remediation. So, it can be achieved in two ways. It can be achieved in two ways. One is in situ bioremediation and ex situ bioremediation. In situ bioremediation and ex situ bioremediation. So, in situ means taking off pollutants, that is a degradation of pollutants where it is, that is within the place, that is called as in situ bioremediation. It is of two categories: intrinsic in situ bioremediation and engineered in situ bioremediation. Intrinsic in situ bioremediation and engineered in situ bioremediation. So, coming to the intrinsic in situ remediation, use of innate capabilities of microorganisms. So, the microorganisms will be having certain innate capabilities to degrade the pollutants. So, we are using such capabilities without any modifications to degrade the pollutant. Such method is called as intrinsic in situ bioremediation. Next, coming to the engineered in situ remediation, that is modification of certain physico-chemical properties of the microorganisms such as pH, nutrient availability and oxygen supply, which can boost up or which can enhance the capabilities of microorganisms, such is called as engineered in situ bioremediation. Next, ex situ bioremediation, it is taking up pollutants from one place to other selected area where there is will go for degradation of pollutants. Such method is called as ex situ bioremediation. Ex situ bioremediation. So, the use of microorganisms in removal of environmental pollutants. So, we use genetic engineering technology to modify the microorganisms to enhance the capabilities to degrade the pollutants. Such we learned in genetically modified microorganisms, that is we call as GEMS. So, genetically modified microorganisms, genetically engineered microorganisms. So, we are modifying the genome. So, it is a modification of genome modification by using rDNA technology, rDNA technology or genetic engineering. Genetic engineering technology. So, the genome will be modified by using rDNA technology or genetic engineering technology to enhance the capabilities of microorganisms, that is called as genetically modified organism, GEMS. So, we have certain examples. Pseudomonas diminuta, parathione, which degrades the parathione. Next, Pseudomonas olivarians, alkanes. Next, Pseudomonas putidium, mono and dichloro aromatic compounds. Next, alkaligens, alkaligens, phenoxic compounds, phenoxy compounds. Next, Acinetobacter, Acinetobacter, chlorobenzenes. So these are some of the modified organisms through genetic engineering technology or DNA technology. Pseudomonas diminuta, which is used to degrade the parathione. Next, Pseudomonas oleovarans, alkanes. Pseudomonas putidea, that is mono and dichloroaromatic compounds. Alkaligens, phenoxy compounds, and Acinetobacter, that is chlorobenzenes. So these are the list of certain genetically modified microorganisms which can act on certain pollutants. So we know that Pseudomonas putidea, which we call as a superbug. So this bacterium is introduced with certain plasmids. So this is the genomic DNA. So this bacteria is introduced with certain plasmid which contains the genes which degrades the compounds. So for example, octagene. So this is the octagene which degrades the octane. If you have XYL gene, it degrades the xylene. CAMP gene, it degrades the camphor. Right. So we are introducing the genes into the bacteria which can perform the degradation process by the expression of the genes. For example, octagene. So if the octagene is expressed, it can degrade the octane. XYL gene for xylene, CAMP for camphor degradation. So we can introduce the gene of interest into the bacteria so that it can gain certain characteristic features to degrade the pollutants. So this is created by Anand Chakravarti. So which can degrade the oil spills. So the oil spills in the water can be degraded by using the such genes, octagene, xylene gene and camphor gene. So this is about bioremediation of pollutants. So removal of environmental pollutants by microbial organisms. Removal of environmental pollutants by microbial organisms is called as bioremediation. So this can be done in two ways, that is in situ bioremediation and ex situ bioremediation. So coming to the in situ bioremediation, so we are degrading the plasmid within the place, degradation of plasmid within the place, that is called as in situ biodegradation. So there are two categories: intrinsic in situ degradation and engineered in situ biodegradation. So intrinsic in situ degradation is use of innate capabilities of the microorganisms to degrade the pollutant, that is called as intrinsic in situ bioremediation. So we are not modifying any microorganisms. Such method is called intrinsic in situ microorganisms. intrinsic in situ bioremediation. Next, coming to the engineered in situ remediation, it is a modification of certain physico-chemical properties like pH, nutrient availability and oxygen supply. So, we are modifying certain changes so that the microorganisms can get certain more capabilities to degrade the pollutants. That is called a engineered in situ bioremediation. Next, coming to the ex situ bioremediation. So, taking up pollutants from one place to the other area, that is selected area, to degrade the pollutants, that is called as ex situ bioremediation. Coming to the microorganisms, so we can modify the genome of microorganisms through RDNA technology or genetic engineering technology, so that it can attain the certain special characteristic features. So, this modification will gain certain characteristic features for the microorganisms. So, the genetically modified organisms can have special characteristic features when compared to other microorganisms. So, there are certain list of microorganisms which is having special characteristic features. So, Pseudomonas diminuta, so which can degrade the parathiones. Next, Pseudomonas olivarians, which degrades the alkanes, and Pseudomonas putidea, which degrades the mono and dichloroaromatic compounds, alkaligens, that is phenoxy compounds, and Acinetobacter, that is chlorobenzene compounds. Next, so we know that Pseudomonas putidea is in superbug. So which is created by the modification of the genome of microorganisms. So where we introduce the desired gene into the bacteria. We have different genes, oct gene, xylin gene, camphor gene, which can degrade the specific components. Oct gene, which degrades the octane, XYL gene, which degrades the xylin, camp gene, which degrades the camphor. And there is a possibility of introduction of 2 genes or 3 genes at a time. So this is genomic DNA. So this is a plasmid, the plasmid which contains oct gene, which contain XYL gene, which contain camp gene. So each plasmid, single plasmid can have more than one gene, so that a single plasmid can degrade the more number of pollutants. So we can adapt any of this method either by using a single specific gene or a combination of specific genes, that we call. So that is through RDNA technology or genetic engineering technology. So this is all about bioremediation of pollutants. So in RDNA technology, there are several certain steps to be followed to create the genetically engineered microorganisms. So steps involved in RDNA technology. First one is selection of microorganisms, species. That is the first step. Second one is modification of plasmid DNA, with desired genes. Third one is introduction of this modified plasmid into microorganisms, and fourth one is expression of these genes. So these are the steps involved in RDNA technology. So the first step is the selection of microorganism species. So the compatibility of microorganisms is selected according to the environment in which it has to be worked. According to that, the microorganisms has to be selected. So, for example, if you take Pseudomonas putidea, so which is having capability to remove the oil spills in the oceans. So, this Pseudomonas putidea can able to grow on water, on oil. So, such microorganisms has to be selected in order to go for modification. That is the first step, selection of microbial organisms, species. Next, modification of plasmid with desired genes. So, whatever our desired genes has to be introduced into plasmid. So, this plasmid again, so that is modification of plasmid with desired genes. Either we can introduce single specific gene or multiple specific genes in a single plasmid. Next, introduction of plasmid into microorganisms, that is the third step. So, after modification of plasmid, this modified plasmid has to be introduced into the microbial organism, so that it becomes genetically engineered microbial organisms. And last step is expression of the genes. So, the plasmid which is having self replicating capacity will undergo replication and will undergo further expression of that genes which is present on the plasmid. That is the fourth step, that is expression of genes. So, the steps involved in RNA technology is selection of microbial organisms, modification of plasmid with desired genes, introduction of plasmid with microbial organisms, and expression of genes. So, this is all about bioremediation of pollutants. So, with this we have completed bioremediation of pollutants.